Okay, welcome everybody to the lab presentation on the nervous and the endocrine system. Um, this material will be covered on the upcoming lab practical, so please be sure not only to listen to this podcast and take notes, but to review the material in your lab manual and in your book as well. So, for the purposes of the practical, you need to be able to identify all the structures on the neuron and brain model, as well as the torso model, and hopefully next week in class we'll do a sheep brain dissection. I'll include a sheep brain dissection at the end of this podcast for you to watch. But it's helpful if you can do it, or if you can watch me do it live as well. We're going to relate function to structure, and we're going to integrate the brain structures with the endocrine system. They're both communication systems in the body. And then we'll finally touch on the relationship between glands, hormones, and effectors. So this is just a list of what you need to know. It's in the PowerPoint up on Angel, so be sure and be able to identify and tell me the purpose of each of these structures. So you want to know the name, the function, and the location. Here you can see the rest of what you need to know regarding the endocrine system and the nervous system. Okay, so make sure and be able to define everything that's on this checklist. So we'll start with the motor neuron model. This is in the lab. We got one out during lab session today, and we were able to identify and locate each of the following structures and also to identify their purpose. So if we start with the neuron itself, this is the cell body or soma of the neuron. It contains the cytoplasm, and it also contains these pigmented granules called Nissl bodies, which are essentially waste components that the neuron has a difficult time getting rid of over time. As we age, one of the things that happens is that these Nissl bodies begin to accumulate, and it eventually begins to impede the function of the neuron itself. And this is why the function of the nervous system tends to decline as we age, because of the buildup of these waste products that aren't effectively eliminated. In the middle of the cell body, you can see the nucleus, which contains the genetic material, and you'll find inside the nucleus a very large nucleolus. And the nucleolus, remember, is the site of ribosomal RNA synthesis, which is important in nerve cells because they do a lot of protein synthesis in order to do their job, which is sending and receiving electrical signals. So because of this intensive amount of translation that's going on, we need to make a lot of ribosomes. The ribosomes are the protein building components in the cell. As a result, the nucleoli are rather large. Sorry about that. Okay, while we're still looking at the cell body, I want you to notice these extensions, these little gray extensions coming off the cell body, and those are called the dendrites. And what the dendrites are kind of like antenna. What they do is they listen to what's going on outside the cell, and what they're designed to do is to respond electrically to chemical signals that come from other nerve cells. So what you're actually seeing here with these little things that look like little rubber stoppers almost, with these little cylindrical extensions coming off of them, those are the axon terminals of other neurons that are coming into very close contact with the dendrites. Although they're not physically touching them, there's a gap called the synapse that separates each of these from their supposed target. And what happens when an electrical signal gets sent is that it travels from the dendrite to the cell body to the gateway to the axon called the axon hillock, and then all the way down the axon hillock until it gets to the axon terminal. These are axon terminals, okay? And at that point, the release of neurotransmitter is triggered when calcium moves inside the axon terminal, and little membrane-enclosed vesicles called synaptic vesicles filled with neurotransmitter fuse with the presynaptic membrane, which would be this end of the axon terminal, and then release the neurotransmitter into the synaptic cleft. Now, the other parts of the neuron that are important to its function include the axon hillock, which is the gateway to the rest of the axon. This is where all of these chemical signals that lead to changes in voltage on the surface of the dendrite or of the soma, on the membrane of these structures, where they will eventually come together, and at the axon hillock a decision will be made by the neuron whether or not to begin the process of generating an action potential or not to fire. If we reach what's called a threshold voltage, what will happen is that sodium gates will open up, and that will begin the cycle of electrochemical events that will produce the action potential, and that will ultimately lead to the movement of the action potential away from the axon hillock and toward the axon terminal. Okay, so if we were to chart the movement of an electrical signal through the neuron, it would look a bit like this. What we would do is we would start with the electrical signal on the surface of the dendrite or on the soma, on the membrane portion. These different changes in voltage would all kind of ripple together just like waves on the surface of a pond. Some of them would cause the cell to tend more towards perhaps firing, reaching the threshold voltage, and some of these graded potentials would cause the cell to become less likely to fire. What will happen is that all of these electrical potentials will eventually flow, just like water flows down a river, to the axon hillock. So thousands and thousands of little tiny electrical signals will approach this region of neuron. And here, if we reach the critical voltage, okay, if we manage to get to the point where we reach what's called the threshold voltage of the axon hillock, that will begin the chain of events that leads to the production and the propagation of an action potential, which will move in this direction. Okay, so you have to imagine this is the axon here. The signal's heading down the axon away from the nerve cell body. It'll continue. This is just a continuation of this long process, heading down, 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 okay, down the axon, until eventually we reach the axon terminal. At the end of this very long process are going to be little extensions that look a lot like these little rubber things here. And at that point, what will happen is that calcium will move into the axon terminal. Neurotransmitter-filled vesicles will fuse with the ends of the axons, and they'll release the neurotransmitter into these little gaps, these synapses, on the next target downstream. It could be another nerve cell, it might be a muscle, it might be a gland. It all depends on what part of the nerve pathway we happen to be looking at. Now, if we look in a little more detail at the axon, what we find is that this is the extension that allows the electrical signal to travel away from the soma and towards the axon terminal. So this is axon, this is all axon. If you look real close on the model, you can see in here neurofilaments and mitochondria. You can also see some Nissl bodies around here, and this serves as a way for the neuron to transport material back and forth from the axon terminal to the soma, and from the soma to the axon terminal. So it's sort of like a set of conveyor belts that allow these things to be moved around inside the cell. Now, wrapped around the axon are these Schwann cells that produce a white lipid type substance called myelin. And what happens is that they sort of wind around the axon at strategic points, and this helps insulate the electrical signal so that it doesn't leak out. But you'll notice that there are little gaps in the covering. These little gaps right here are called nodes of Ravnier. Okay, I'll circle a couple of them there. And what the nodes of Ravnier serve to do is accelerate the electrical signal, because what it'll do is it'll actually hop from node to node to node when we're talking here about an electrical signal. So what'll happen is the electrical signal will actually shoot very quickly underneath these insulated parts of the axon, and that will lead to a series of events that rapidly regenerates the action potential. Here, say at this node, that'll open up sodium gates, cause the electrical signal to shoot real quick under the next the next insulated part of the axon, the next part that's shielded by the myelin sheath, and then that'll set into motion another series of sodium gates opening up, and the action potential as a result will seem to hop from space to space to space. And so this can get very fast. This can get up to 200 miles an hour inside a motor neuron, some of the fastest electrical conduction that you see in the human body. Now, not all nerve cells have a myelin sheath around them. Some of them are what we call unmyelinated, and as a result, their nerve signals move much more slowly, about 20 times more slowly, in fact, than that of a myelinated neuron. And the reason we need different speeds is because the body has different demands for electrical information. Sometimes the demand for electrical information is immediate, such as motor commands that have to get to where they're going right away. And sometimes we don't need that kind of speed in an electrical signal, so we don't require the myelin to speed up the electrical signal going from the soma to the axon hillock to the axon all the way to the neuron terminal. Now what you're looking at here is a model of a human brain, and next to it an actual preserved sheep brain, so that you can compare some of the important structures. So what I want you to notice are you can see here the different lobes of the brain, which are named for the bones of the skull that cover them. Okay, so for instance, the frontal lobe is underneath the frontal bone. The temporal lobe is underneath the temporal bone. The parietal lobe back here is underneath the parietal bone, and the occipital lobe is underneath the occipital bone. Now, you might ask, okay, great, what do these lobes do? Well, the frontal lobe is the part of the brain that's involved with our higher thinking, judgment, and problem-solving skills, and it's also where we plan voluntary activity. This is where we decide how it is we want to do things like walk across the room, or bend over and pick up a piece of paper, or to write a letter to somebody. These motor commands are assembled in the frontal lobe, and then they're actually sent to an area of the brain right about here, okay, this little groove, this little ridge that I'm tracing, and that's called the motor cortex, okay? So the plans get hatched here, and they get sent to the motor cortex, and what happens next is that these finalized plans will be sent via white matter, which are the signal transmission portions of the brain, to the brain stem, and from there to the spinal cord, and from the spinal cord they'll head out to the peripheral nerves, which will eventually carry the electrical signal all the way to their target. Now, in the case of the somatic motor system, the targets are skeletal muscle. In the case of the autonomic nervous system, they could be glands or other tissues. Okay, so just sort of a general idea of what the frontal lobe is all about. This little ridge here, the motor cortex, sits right in front of this little groove right here, which is called the central sulcus. So let me draw a line right across the central sulcus there. Okay, and then there's a groove of tissue right behind it, which is called the sensory cortex. Okay, this, I'll draw another line here. Okay, and the little ridge of tissue that runs behind the central sulcus is the postcentral gyrus. So actually, let me be a little bit more clear about that. If we redraw this here, we can see right there. Okay, so let me redraw these just a little bit. So this is the central sulcus right here in green. Okay, the motor cortex is right here in red. Okay, and the sensory cortex, which marks the beginning of the parietal lobe, is right here in blue. Okay, so that's the beginning of the parietal lobe. That right there is the frontal lobe. Now, the parietal lobe, which is behind the frontal lobe, is responsible for receiving sensory information from all over the body and then sending it to what we call the sensory association area of the brain, which is basically all through here. And what we do in that part of the brain is we assign a category to the sensory information. We decide whether it's light or sound or pressure or what have you. And from there, that information is relayed to other parts of the brain so that we can decide what we want to do about it. So that's a basic job description for the parietal lobe. The temporal lobe, right here, okay, is responsible for interpreting our auditory information. Okay, our sense of hearing and balance comes from electrical signals that are sent to the temporal lobe. And then back here, the occipital lobe, which is at the back of the brain, draw that here, okay, that is responsible for interpreting our visual information. Okay, so occipital, temporal, parietal, and down here in the frontal. The cerebellum, which is this funny looking thing under the occipital and temporal lobes, is responsible for synchronizing what we're doing with what we want to do. Okay, so it is our on-the-fly motor coordinator. And it looks in a real brain just like turkey burger, and it has grooves and ridges in it just like the cerebral cortex does, and that's again for the purposes of increasing the surface area of the brain so that it can do a lot of electrical work. Okay, so anytime you see a convoluted surface or anything that's designed to increase the square footage that a particular tissue covers, it's generally because it has to do a lot of biochemical work in a very tiny physical space, and that's true for the cerebral cortex and for the cerebellum. Now, this little white stalk down here, that is the brainstem, and the brainstem has three parts. This is the medulla oblongata, this is the pons, and then above the pons you would find the midbrain, and then deep inside, underneath the cerebral cortex, you would find the diencephalon, which contains the epithalamus, the thalamus, and the hypothalamus, which serve as the gateway to allow stimulation to get to the cerebral cortex. So it's sort of like the brain's sensory filter, if you will, because we don't want to be totally aware of everything that's going on around us all the time, because some of that information isn't useful. You can see down here with the sheep brain that you've got a lot of the same basic structures, but they're present in slightly different dimensions. But you can see here the sulci and the gyri, the cerebral cortex, the cerebellum, and you can see the parietal and the frontal and the temporal lobe and the occipital lobe back here, and of course the brain stem. And hopefully next week in class we'll actually get to take apart a preserved sheep brain, and I can show you some of these structures a little bit more clearly. If we look at the underside of the brain, what we see is several structures that we want to be aware of in the brain stem: the medulla oblongata, the pons, the midbrain. This is the pituitary gland, and this right here is the optic nerve, and this is the olfactory bulb. This is cranial nerve number 1. This is cranial nerve number 2. And the pituitary gland is essentially the boss of the rest of the endocrine system, and it receives its instructions from a part of the brain called the hypothalamus. You can also see down here the cerebellum, okay, and this will be the beginning of the spinal cord. Now in the sheep brain you can see some of these structures as well. There is the medulla oblongata, the pons, and up there is the midbrain. There's the optic nerve. There's the optic chiasm. Chiasm means to form an X. So this is where the optic nerves cross from left to right and right to left and go into the back of the eyeball. And you can see here the olfactory bulbs, which sit just underneath the frontal lobe and right on top of the ethmoid bone, and this provides our sense of smell. And here you can see the sheep brain in a sagittal section, and you can pick out these different structures. You can see here the diencephalon, the midbrain, the pons, and the medulla oblongata. There's the cerebellum right here. This is the corpus callosum, which is a tract of white matter that connects the two hemispheres of the brain. You can see here the cerebral cortex, and you can also see the occipital, the frontal, and the parietal lobes as well. Now if we take a closer look at the sheep brain and the human brain model, we can pick out the frontal lobe, okay, indicated here by the number 1, the parietal lobe back here in the sheep brain and here in the human brain model, the occipital lobe back here, okay, and back here in the sheep brain. And you can also see the cerebellum that has 2 layers to it. An outer layer called the folia, which are these deep grooves of gray matter on the surface of the structure, and then this little tree-like white structure called the arbor vitae. That means tree of life in Latin. And this set of white matter are what brings in information to the cerebellum and sends the information out from the cerebellum at the same time. Over here you can see the thalamus, okay? The thalamus is the gateway to the cerebral cortex, and if we were to remove the brainstem and the diencephalon from the cerebral cortex, what it would look like is a double-headed mushroom with 1 stalk. Okay, so if you can imagine that in your head, that would be exactly what the thalamus looks like if we remove it from its overlying structures. The hypothalamus is this area just above the pituitary gland, which is right here. Okay, so you can see here the hypothalamus and the pituitary gland. The hypothalamus is the boss of the pituitary gland, and the pituitary gland is the boss of the rest of the endocrine system. The midbrain is indicated here by 7. It's that piece of tissue that connects the pons to the diencephalon, and you can also see the pons down here, this little bulge indicated by 8, and it's a place where a lot of major nerve tracts run through the brainstem up to the diencephalon, and from there up to the cerebral cortex. The medulla oblongata is an area where a lot of nerve fibers cross from the left to the right side of the body and from the right to the left side of the body at structures known as pyramids, although they don't look like pyramids if you were to look at that part of the brainstem from an anterior aspect. They look like a pair of baseball bats lying side by side on the front of the brainstem. Okay, so those are the pyramids of the medulla oblongata. You can see them here in the model, and you can see them here in the sheep brain. You can also see here a couple of endocrine glands that are very closely associated with the brainstem. The pituitary gland sits inside a little depression in the sphenoid bone called the pituitary fossa, so it's very well protected, which is important because it is the commander-in-chief of the rest of the endocrine system. And then about 180 degrees opposite the pituitary on the back side of the brainstem is the pineal body, which secretes a hormone called melatonin, which controls when we're awake and when we're asleep. And that's important because that enhances our survival in the wild. We don't want to be active at times of the day where there's minimal light because we can't see what we're doing, and we're likely to injure ourselves accidentally or get eaten by a predator. And we want to be active during the time when there's light out because we can see, to gather food, to find shelter, and to avoid being eaten. So this is a very primitive survival response. We think this is related to these activity levels because what scientists have found is that levels of melatonin are high at night and they tend to be low during the daytime. Okay, we'll pick up with the second part of this presentation in just a moment. Okay, we're going to pick up with a further discussion of the organization of the nervous system, and what we want to talk about is how information flows from sensory receptor to integration point to output. So essentially what happens throughout the body is that we have specialized nerve cells that are designed to transform physical stimuli into electrical information, and they can respond to any number of different physical changes such as light or sound or pressure or pain or temperature and so on. They're located all over the body. A lot of them are in your special senses, for instance, photoreceptors in the back of your eye. You've got chemoreceptors in the nasal cavity and in the oral cavity. You've got thermoreceptors in the skin that respond to changes in temperature, nociceptors that respond to pain, pressure receptors, all sorts of specialized cells that do nothing but change the information that's out there in our environment into something that our nervous system can understand and interpret. So the sensory input goes from sensory receptors to specialized cells that are called neurons that we talked about in the last block. The neurons carry that electrical information from the extremities of the body, okay, the part that interacts with the environment, what we call the peripheral nervous system, to the central nervous system, which is the brain and the spinal cord. So these afferent neurons, afferent means incoming information, head into the spinal cord, and they actually enter the spinal cord at the back. An area called the dorsal root is what carries sensory information into the spinal cord, and then from there ascending pathways climb up to the brain, and then the brain decides what it wants to do about that sensory information, and what it'll do is generate now a plan of action that will be sent from the brain to the spinal cord and then out the peripheral nerves to motor neurons, which talk to things like skeletal muscle, smooth muscle, glands, other tissues, in order to produce a response. Okay, so sensory input, then we interpret the information, then we form a plan of action, and then we send that plan of action down the spinal cord to the peripheral nerves, and then the effectors, the things that carry out the commands, do something about the perceived stimulus. And so here you can see a classification of the different anatomical divisions of the nervous system. The central nervous system made up of the brain here in the spinal cord. So this is all central nervous system tissue. And the peripheral nervous system is everything outside the central nervous system. So that includes the spinal and the cranial nerves and the sensory receptors. And this is how the electrical signal moves in and out of the central nervous system. So you can see here the yellow box indicating the central nervous system structures, the spinal cord, the brainstem, the brain, and the purple trapezoid here indicating the peripheral structures, the spinal nerves, the cranial nerves, and the receptors. Now if we look at a neuron as a single structure, which is a little bit more clear than perhaps the models we were looking at earlier, what you can see is that the soma or cell body of the neuron contains the nucleus, which holds the genetic information, and also the cytoplasm, which surrounds the nucleus, contains lots and lots of ribosomes and cellular organelles and what we call biosynthetic machinery, which is there to build and transport proteins to different parts of the nerve cell. These little blue staining areas, as we learned in the last installment, are what are called Nissl bodies, and these are basically waste products that build up inside the cell over time and eventually will impede its function, but it's the sort of stuff that the neuron has a difficult time getting rid of due to the fact that it's it's difficult to move outside the cell. So these are what are called inclusions. These little extensions that come off of the cell body, those are called dendrites. And what dendrites do is they monitor the external environment for chemical and electrical changes. They have all kinds of receptors on their surface, as does the nerve cell body, which will bind to special chemicals called neurotransmitters, which will open ion gates on the membrane and will cause the membrane potential, the voltage at that part of the membrane, to change. Some of these chemical signals will tend to cause the neuron to become more likely to produce what's called an action potential, and some of them will cause the neuron to become less likely to produce an action potential. What happens is these electrical signals coalesce in the soma, the membrane of the soma, the nerve cell body, is that eventually they'll come to a very special place in the nerve cell body called the axon hillock, which is basically the gateway for electrical signals to move from the nerve cell body down the rest of the axon and ultimately to the axon terminal, where it will cause the release of a neurotransmitter. But before that can happen, we have to get to what's called a critical threshold voltage. If we don't do that, if we don't reach this critical voltage, what will happen is that the nerve cell will not generate an electrical signal that will travel down the axon and result in the release of neurotransmitter. And so it'll basically be unresponsive. The signal will stop essentially with this neuron. But if we do reach what we know as the threshold voltage, what will happen is it will set into motion a series of events, a series of changes in the nerve cell membrane that will result in an electrical potential being propagated from the axon hillock all the way down to the axon terminal. And that happens as a result of the opening and closing of ion gates in a very precise fashion. If you refer to the lecture on the nervous system, there are a couple of videos that talk at length about how membrane potential is established and how an action potential is propagated. So, make a long story short, essentially, when we're talking about the movement of a signal through the axon, we're starting here at the dendrites, we head to the axon hillock, and then we head down, down, down the axon until we get all the way to the axon terminal, okay, and then we release neurotransmitter. So I'll draw a big NT here for what happens when the electrical signal gets to the end of the axon. So it only flows in one direction. That's a very important thing to remember. Electrical signals cannot back up and go back towards the cell body. They can only proceed from dendrite to soma to axon and then all the way to the axon terminal. Now you'll notice in this little inset here showing you a micrograph of an actual neuron. You can see here the axon, you can see the dendrites coming off, okay, and what you'll notice is that the neurons are very seldom all alone, okay. They tend to be surrounded by other neurons or other targets, and so these things are in very close proximity to each other, and that's important because when the electrical signal does reach the end of the nerve cell, the neurotransmitter, which is going to carry the electrical signal from one nerve cell to the next, doesn't want to have to travel very far in order for that to happen. What it does is it diffuses across a little tiny gap called a synaptic cleft, a little opening between one neuron and another neuron, or one neuron and a target, and it will bind to receptors on the next nerve cell, and then that electrical signal will head further down the nerve pathway. Now if we look at a typical neuron, what you see is there's a lot of extensions that come off the cell body. These are the dendrites, and then the one long extension, that's the axon. In motor neurons, the nerve cell body and the dendrites are located in the central nervous system in the gray matter. We'll look at a cross-section of a spinal cord in just a bit, and what you'll see is that there's a butterfly-shaped brown mass, in the center of the spinal cord, and that's where the nerve cell bodies and the unmyelinated axons and the dendrites sit. The white matter is myelinated axons, and in the motor neuron, the axons extend all the way to the target, which in the case of a motor neuron is a skeletal muscle cell. Okay, so when the electrical signal gets to the end of this long process and gets to the axon terminal, what will happen is that that change in voltage will result in the opening of calcium channels. The calcium will move into the axon terminal, and that will trigger the release of little membrane coated vesicles that contain neurotransmitter fusing with this membrane and releasing their contents into the synaptic cleft, whereupon the neurotransmitter will drift across this tiny gap, bind to the postsynaptic membrane, and change the electrical signal in the next cell in the pathway, whether that be another neuron or perhaps some other type of target tissue. Now, one of the things you might well ask is why isn't the myelin sheath in the axon of the motor neuron a continuous sheath? And the reason for that is that when electrical signals move from the axon hillock to the axon terminal, due to the structure of the myelin sheath, what will happen is that the electrical signal will actually hop from one node to the next very, very rapidly in a process known as saltatory conduction. And these speeds can reach up to 200 miles an hour in motor neurons. And the reason for that is that motor output needs to be very quick in order for it to be useful. So what will happen is that this electrical signal will fly from one end of the axon to the next and then ultimately reach the axon terminal, and that will result in the release of neurotransmitter. If the entire axon were covered by a myelin sheath, we wouldn't be able to produce a change in the membrane voltage as we move from the axon hillock down to the end of the axon terminal, because we would have insulated the cell membrane from the outside environment, which means we wouldn't be able to move ions from the outside of the cell to the inside of the cell. So this is why we have these periodic gaps in the myelin sheath. Remember that each of these myelin segments is one Schwann cell, and the myelin serves as an insulator. Okay, so it allows the movement of electrically charged particles underneath these portions of the axon to happen very quickly, and that's just enough distance to get to the next node in the axon pathway and produce the voltage change that will ultimately cause sodium gates to open up and change the membrane potential at each node, which will then shoot under the next myelinated portion of the axon, and that's why the electrical signal will be seen to hop very quickly from node to node to node, because the movement under the insulated part of the axon is very quick compared to the voltage changes that take place at each of these nodes. So this is what helps to speed everything along. If the entire axon were covered, what would eventually happen is that the movement of charged particles through the axon would only go a certain distance, and then it would stop, and as a result we wouldn't get the electrical signal to the end of the cell. And what you're looking at here is just a sort of a description of what constitutes a fast versus a slow neuron. So remember that a larger diameter axon is going to be faster, and the reason is that there's less resistance to the movement of charged particles. When you've got a lot of space to work with inside the cell, you could almost think of it in a way very similar to the movement of fluid through a large diameter pipe versus one that has a very tiny diameter. And also, when an axon is myelinated, the signal transmission rate is going to be faster than an unmyelinated axon. So alternate areas of exposed axon membrane are covered with this myelin, and the covering helps to speed the electrical signal along. So what will happen is the signal will start at the beginning of the axon. The changes in voltage will get to the point where it reaches the insulated part of the axon covered by the myelin sheath, and it'll shoot very rapidly through this part of the myelin sheath, and then that change in voltage will be reproduced here, and the same thing will happen. So change in voltage, shoot, change in voltage, shoot, change in voltage, shoot, and that's what allows the electrical signal to seemingly hop very rapidly from node to node to node until it gets all the way to the end of the axon. If we look at the way that axons become myelinated, in the peripheral nervous system, it's the Schwann cell that does this job, and what it'll do is it'll wrap around the axon in a jelly roll-like fashion and insulate that portion of the axon from the outside environment. So you can see how we begin producing the myelin sheath here, and when we're done, what we have is an actual independent cell that's filled with myelin that has rolled over upon itself. This is cell membrane, and then the myelin is actually inside the Schwann cell, and then this wraps itself around this extension, the axon, that comes directly off the soma, the cell body. And this is what helps to speed electrical conduction along. There are diseases in which the myelin sheath can be broken down, it can start to die, and as you can probably guess, those diseases lead to difficulty in the neuron sending its signal from point A to point B. An example is multiple sclerosis, where the immune system begins to attack the myelin sheath and destroy it. The result is that the electrical signal, when it gets to the axon, does not have enough push to it to get to the next node, and as a result it tends to die out before it reaches the axon terminal, and the result is that we can't produce an electrochemical signal near the target. And so in multiple sclerosis, what happens is that voluntary muscle movement becomes difficult. Eventually the muscles will atrophy because of lack of innervation, and at that point we can't use that muscle group anymore. There are other diseases that affect the myelin sheath adversely, and they all result in the same thing, which is that the electrical signal doesn't get to where it's supposed to go in a timely fashion. Now, if we look at the central nervous system, if you refer to the lecture on the central nervous system where we talked about how development occurs, remember that we touched on the fact that the neural tube is basically a structure that drops below the layer of the ectoderm at about 10 to 12 weeks, and that eventually is going to form the spinal cord, the brainstem, and the brain. The neural tube actually folds up from the lumbar region up towards the cephalic region, up towards the head, in sort of a stepwise fashion, and there are open spaces that are left in both the spinal cord and the brain through which cerebrospinal fluid will flow. The brain, once it fully develops, is going to have a total of 4 ventricles, 2 lateral ventricles, a third ventricle between them, and a fourth ventricle below the third ventricle that sits in between an area of the brainstem called the pons and the cerebellum. And that fourth ventricle will be connected to a continuous canal that runs down the middle of the gray matter of the spinal cord called the central canal, and that serves as a route for the cerebrospinal fluid to flow both through the inside and outside of the brain, and also bathe the inside and outside of the spinal cord as it escapes little apertures in the fourth ventricle that are located on the lateral and medial side. And it's important to keep the cerebrospinal fluid constantly being produced and being reabsorbed so that we can maintain the proper pH and body temperature and electrolyte concentration and oxygen concentration around the neurons that do a lot of very rapid work and need a constant supply of oxygen and nutrients in order to continue to function. So the neural tube becomes the brain and the spinal cord. The opening in the neural tube becomes the ventricles, and remember there are 4 ventricles in the brain. They're filled with cerebrospinal fluid. Um, where does the cerebrospinal fluid come from? It comes from the choroid plexus that sits in each of the ventricles, which is the site where the blood plasma is filtered to produce the cerebrospinal fluid. So it's actually a blood filtrate. And then you might ask, well, okay, where does the cerebrospinal fluid eventually go once it's been used, once it flows around the brain and the spinal cord? And the answer is that it rejoins the circulation at special points stationed in the arachnoid mater, one of the membranes that surrounds the brain and spinal cord, at spots called arachnoid villi. What will happen is that the CSF will diffuse through those protrusions, rejoin the venous circulation, and eventually return to the heart. Okay, so we make the CSF in the ventricles. It flows through the inside and outside of the brain and spinal cord, and eventually it rejoins the circulation and the arachnoid granulations, and as a result contributes to blood volume. And eventually what will happen, of course, is that the flow of fluid through the body will be completed as the fluid exits the body through your sweat and through your urine and through your breath. Okay, so you have to keep that flow of fluid constantly moving. If you have an obstruction in the movement of this cerebrospinal fluid through the brain and spinal cord, it results in an increase in pressure in either the vertebral canal or in the cranial vault. And if that happens, it can kill brain cells. That increase in pressure can kill the neurons. Remember that in the central nervous system, neurons do not repair or reproduce, and so that loss is permanent, and the result can be coma and death. Okay, so very important that we realize that a free flow of CSF is important to maintain the health of the brain and the spinal cord. In infants, because the flat bones of the skull have yet to fuse, what we see when the cerebrospinal fluid flow is restricted is an increase in intracranial pressure that causes the cranial vault to enlarge so that the head looks a little bit like a light bulb. And the reason that this isn't a lethal situation in infants is because the vault can expand to accommodate that intracranial pressure. But in order to fix the situation, what we have to do usually is put a shunt, a bypass, running from one of the ventricles in the brain that's upstream of the block, and then drain it into the abdominal pelvic cavity. And this allows the fluid to rejoin the circulation, and as it gets reabsorbed by the capillaries that permeate the peritoneum, which is the membrane that lines the abdominal pelvic region, and that allows it to rejoin the circulation. What will eventually happen is that the intracranial pressure will drop, the cranial vault size will go back to normal, and normal function will be restored. And so here you can see the adult brain fully formed. If we work up from the spinal cord, we can see the medulla oblongata, the pons, and the midbrain, and this is the diencephalon, which is made up of the epithalamus, the thalamus, and the hypothalamus, which is attached by the infundibulum right here to the pituitary gland. And then the cerebellum sits right behind the pons and underneath the occipital lobe of the brain, while the cerebral cortex surrounds the diencephalon, and it's where most of the integration of signal input from the peripheral nervous system takes place. Now, one of the things that you want to notice just looking at the brain before we get really deep into the structure is that it's really wrinkly, okay? And that those wrinkles are called sulci and gyri. The gyri are the protrusions, and the sulci are the deep grooves in the brain. And the reason for these modifications to the surface texture of the brain is because the brain has a lot of work to do. It sends and receives electrical and chemical signals constantly, and so it needs space to do that. And what these grooves and ridges do is they increase the amount of space available to do that work. Now, if we break down the brain a little bit further, what we see is that the cerebral hemispheres are broken up into lobes that are named for the cranial bones that sit on top of them. So this is the frontal lobe, the parietal lobe, and the temporal lobe. Back here is the occipital lobe, and then there's a fifth lobe that's actually between the temporal and the parietal lobes. You have to pull them away to see it, which is called the insula. If we look at a cross-section of brain tissue in detail, what we find is that the gray matter of the brain is on the outside and the white matter of the brain is on the inside. Remember that gray matter is composed primarily of nerve cell bodies, and that white matter is composed primarily of myelinated axons. And so remember, when you see gray matter, that's always signal processing, and where you see white matter, that's always signal transmission. The youngest part of the brain, as it turns out, is actually the closest to the surface. The way that the brain forms as it advances from neural tube all the way to completed central and peripheral nervous system is that the neurons deep inside the brain actually crawl up the glial cells, and when they get to the brain surface, they stop and they change into fully functioning nerve cells and start doing their job. And then younger neurons actually climb up above them and lay on top of them and differentiate and continue to engage in electrochemical activity. So unlike a tree, the youngest part of the brain is on the outside and the oldest part is deeper down. Fissures divide the cerebrum into the different lobes, as we noted earlier, and you can see them here in an actual human brain diagram. The frontal lobe in blue, parietal in red, the occipital in purple, and the temporal in green. And they all do slightly different jobs. Okay, so if we were to give a general job description for each lobe of the brain, the frontal lobe would be our seat of problem solving and higher reasoning, and also a lot of our personality is housed in the frontal lobe. It also contains the motor association area and the motor cortex. So this is where plans of action are hatched in the brain, where they are sent to a very special structure called the precentral gyrus, this little ridge of tissue right here that serves as the border between the frontal and the parietal lobe. And from there, those plans of actions are sent to the brainstem, to the cerebellum, and then down to the spinal cord, and then out the peripheral nerves to the effectors, and then we do something. The parietal lobe contains the postcentral gyrus, which we call the sensory cortex, which receives information from all over the body, and the sensory association area, which assigns categories to this different information. And from there, that information is going to be routed to parts of the brain that will figure out what to do about this sensory input. The occipital lobe receives and processes information about vision, and the temporal lobe receives and processes information about hearing and balance. There's other functions, of course, in these lobes, but this is just a sort of a general job description. And while we're at it, we should talk a little bit about the cerebellum, whose job it is to coordinate what it is you're intending to do with what it is you're actually doing. And it edits that information on the fly so that your movements are purposeful and they accomplish the objective that's set by the instructions sent from the frontal lobe. And of course, anterior to the cerebellum is the brainstem, the three parts, of course, the midbrain, the pons, and the medulla oblongata, which lie underneath the thalamus, and the diencephalon, the epithalamus, the thalamus, and the hypothalamus. The main job of the thalamus is to serve as the gateway to the cerebral cortex. So what happens when information comes in from the receptors to the spinal cord to the brainstem and gets up to the thalamus is that the thalamus decides what of that information is going to make it to our conscious awareness and what of that information is going to stay at the level of the diencephalon, and it'll still be acted on, but we won't be consciously aware of it. So the thalamus's job, in a sense, is to kind of pick and choose what information in our environment is important and only send that information to the brain for us to act on. Okay, now this is just another look at the brain showing you some of the more details of the lobes that we just talked about. You can see here that the primary somatic sensory area, which is located in this little blue ridge of tissue that's against the boundary between the frontal and parietal lobe, gets impulses from the body's sensory receptors and it will then send them to the sensory association area where the impulses, the sensory information will be categorized, and it'll be assigned um, a, a category based on uh, whether it's physical sensation of sight or sound or general senses, and then that information will be transmitted to parts of the brain where we can deal with the sensory input. The primary motor area sends impulses to the skeletal muscle, and it's located in the frontal lobe, and you can see this little ridge of tissue here. That is the primary motor cortex. That's where completed commands are sent to the brain, and from there they're going to be relayed to the brain stem, to the cerebellum, and then down the ventral side of the spinal cord, where they'll exit to the peripheral nerves and cause eventually muscle movements based on what these commands instruct the muscles to do. Broca's area is involved in our ability to speak, and you can see Broca's area here deep in the frontal lobe. It's basically motor commands that are directed to the lungs, the muscles that move the vocal cords, the lips and the tongue, all involved in the production of speech. So we have general descriptions for the jobs that these different lobes of the brain do, and if we were to focus in on the primary motor cortex and the sensory cortex, one of the things that we would find is that the amount of surface area of brain dedicated to different parts of the body is going to be disproportionate to our normal body dimensions. So if we look at the motor cortex, what we find is that most of the surface area of the brain is dedicated to motor control of the lips, the tongue, the teeth, the facial muscles, the hands, and much less is dedicated to control of motor functions in the rest of the body. And this makes sense because we need greater motor control over these areas of the body to perform the delicate movements associated with things such as speech and writing and handling and manipulating tools and so on. On the postcentral gyrus, which is what we know as the somatosensory cortex, what we have are different areas of the brain that are dedicated to receiving sensory input from different parts of the body. And so you can see here that the teeth, the lips, the tongue, the face, the hands, and the feet are overrepresented, whereas the rest of the body is represented in a relatively minor way. And that is because there are more sensory receptors in these parts of the body than there are in other parts of the body. And so when we look at these dimensions, what we find is that they will form a very unusual looking human body if we put these together, called the sensory and the motor homunculus. So let's take a look at what that looks like. And here you can see the motor homunculus on the left and the sensory homunculus on the right. And notice that the proportions on the bodies of each of these are going to represent the amount of surface area on the cortex of the human brain that are dedicated to each of these functions. So in the motor homunculus, notice the large face, the lips, the tongue, the very large hands, because of the amount of motor control that we need over those components in the body. Whereas with the sensory homunculus, we see disproportionately large face, lips, tongue, hands, and also feet that are larger in proportion to the rest of the body as well. And that represents the differential distribution of sensory receptors in these parts of the body and the disproportionate amount of surface area of the brain that's dedicated to input coming from those parts of the body as well. This is just another look at the map of the brain showing you some of the different areas dedicated to different functions. We talked about the frontal lobe and the primary motor cortex and the motor association area, Broca's area. We've talked about the primary somatic sensory cortex and the sensory association area. Other areas we want to note: language comprehension, which lies in the frontal lobe, the olfactory area, which lies just on the floor of the brain, right underneath the frontal lobe. It's part of a set of structures in the brain known as the limbic system, which basically assigns emotional content to certain sensory stimuli. And we've got the auditory area in the temporal lobe, the visual area in the occipital, and the gustatory area, our sense of taste, located in the parietal lobe on the lateral aspect of the somatic sensory cortex. Areas that are used for interpretation of different complex stimuli include the language comprehension area in the frontal lobe, the comprehension of speech and language located here in the parietal and temporal lobes, and general interpretive areas located in the parietal and temporal lobes as well. This is what we call the somatosensory association area, where we assign categories to different types of stimuli as they come into the brain from the peripheral nervous system via the spinal cord and the brainstem. Gray matter is the outer layer of the cerebral cortex, and it's made mostly of neuron cell bodies, and underneath it is white matter that's made up mostly of myelinated axons and is involved in signal transmission. So remember, gray matter signal processing, white matter signal transmission. And so you can see the gray matter here. You can also see some layers of deeper gray matter, okay, located inside the white matter. These are the what we call the lentiform nuclei, or sometimes if we group all these together, we call them the basal ganglia, and they modify the nerve input that's coming from the cerebral cortex before it gets sent on to the brainstem and the cerebellum, and ultimately down to the spinal cord and to the peripheral nerves. And you can also see the different nerve fiber tracts that carry information to different parts of the brain, and also ultimately to the spinal cord, to the peripheral nerves, and ultimately to the effectors. So here, if we look, we can see commissural tracts that carry information between the two hemispheres of the brain, and then projection tracts that carry information from the brain down to the brainstem, the cerebellum, and the spinal cord. Notice that they cross here at the level of the medulla oblongata, so the right half of the brain controls the left half of the body and vice versa, and the same is true for sensory information. Sensory information coming in from the left side of the body goes to the right cerebral hemisphere and vice versa as well. This is called decussation, or crossing over, and we honestly don't know why this occurs, but we just know that in higher mammals that's the way that the wiring is set up. There are also what we call association tracts that run between masses of gray matter in the same hemisphere, and they help to move information around from one part of the brain to the other. The diencephalon sits on top of the brainstem and is surrounded by the cerebral hemispheres and is made up of the thalamus, the hypothalamus, and the epithalamus. The epithalamus is the topmost, most superior structure, the thalamus underneath that, and the hypothalamus underneath the thalamus. And then below the hypothalamus is a stalk called the infundibulum, and below that hangs the pituitary gland. So you can see the hypothalamus here, the infundibulum, and the pituitary gland below it. The pituitary gland is governed by the hypothalamus, and the pituitary gland governs most of the rest of the endocrine system through the production of a class of hormones called tropic hormones, which essentially are chemical messages that are put into the blood that make it to different endocrine organs and cause them to produce their hormonal secretions. And so this is why the pituitary gland used to have the nickname the master gland, but it turns out really that the gland that's in charge of the entire endocrine system is in fact the hypothalamus, because it governs the function of the pituitary. You can see also the three structures of the brainstem: the medulla oblongata, the pons, and the midbrain. And then the cerebellum, our coordinator of intentional actions with actual action. If we look within the brainstem, we find a very important structure called the reticular activating system. And what it does is it allows certain stimuli to make it to our conscious awareness via the thalamus. So what happens when we begin to wake up, for instance, from a long night's sleep, is that the reticular activating system is allowing certain stimuli to get to the thalamus. It's also stimulating the thalamus to allow certain information to get to our conscious awareness, and as a result we become more interactive with the environment around us, and we begin to wake up. So the thalamus surrounds the third ventricle. It serves as the gateway to the cerebral cortex by transferring impulses to the correct part of the cortex for localization and interpretation, as well as serving as kind of a wake-up call for the rest of the brain. Individuals that have trauma to the reticular activating system are often rendered in a comatose state. The hypothalamus sits underneath the thalamus and is an important autonomic nervous system center. It helps to regulate body temperature, control water balance, and regulate metabolism, and it also governs the function of the pituitary gland. It's also an important part of the limbic system, which assigns emotional content to different stimuli. As an example, you might smell a flower and comment that that flower smells pleasant. Well, the pleasant is an emotional category attached to that stimulus by the action of the limbic system. If we didn't have limbic input, we would simply say that that smells like a flower, and we wouldn't be able to evaluate whether that is an emotionally pleasing or an unpleasant experience. The midbrain is made up of tracts of nerve fibers that send and receive sensory information to and from the thalamus. These fiber tracts are called cerebral peduncles. You can see the midbrain here. On the back of the midbrain, you've got two bulges called the corpora quadrigemina, and that's Latin for four bodies. And what the corpora quadrigemina does is it receives and interprets information from the visual senses and from the inner ear, okay, which provides our sense of sound as well as our ability to maintain our equilibrium, meaning our ability to stand up straight, to walk and move without falling over. The superior colliculi receive and interpret visual information, that's this bump up here, and the inferior colliculi receive and input auditory information. And then while we're looking at the midbrain, we should also comment on the fact that the pineal body, which is part of the epithalamus, lies just above the corpora quadrigemina, and it's involved in the secretion of melatonin, which is thought to be responsible in part for our sleep-wake cycles. Now, the pons is part of the brainstem. It's the bulging central portion that's composed of fiber tracts and includes nuclei that are involved in the control of your respiratory rate. What they do is they periodically innervate the diaphragm when we're unconscious so that breathing can continue even when we're asleep. Notice that it sits in front of the fourth ventricle and the cerebellum, both of which are surrounding here the fourth ventricle of the brain, which receives cerebrospinal fluid from the third ventricle via this little passageway called the cerebral aqueduct. Notice again the pituitary gland, the hypothalamus, the thalamus, and of course up here what we have is the corpus callosum. Those are the commissural tracts that allow the left and the right half of the brain, the two cerebral hemispheres, to communicate with each other so that we can perform coordinated activity. The limbic system, if you're interested, consists of this little ridge here called the cingulate gyrus, the hippocampus, which you can't really see in this particular diagram because it's been cut away, the olfactory tract in the bulb, and the amygdala. And this is our emotional brain. This is what assigns an emotional content to different stimuli. The medulla oblongata is an important control center for heart rate and respiratory rate, as well as reflexes such as vomiting. It also regulates blood pressure and swallowing. It's the lowest part of the brainstem. It merges into the spinal cord, and it includes a very important set of fiber tracts that cross from right to left and left to right. So this is a major site of decussation. If you were to look at the medulla oblongata from the front, it would look like it had a pair of miniature baseball bats right next to each other on the anterior aspect. Those are called the pyramids, and those are the sites where the fibers actually cross from right to left and left to right in a large number of motor and sensory pathways. The cerebellum lies behind the fourth ventricle and under the occipital lobe, and it has two hemispheres. It has a lot of convolutions in the surface. These little ridges and grooves are called folia, and then deep inside the cerebellum you've got this white tree-like structure that's called the arbor vitae, which is Latin for tree of life. And what it does is it coordinates our intended actions with our actual actions, so it makes sure that those two things match up. And if it doesn't, what it'll do is it'll modify motor output in order to adjust the commands that are coming from the cerebral cortex before it sends them on to the brainstem, so that we do in fact what we are intending to do. And these adjustments happen so quick that we're really not aware of them. In individuals that have damage to the cerebellum, movements are often purposeless and not well controlled. They tend to be rather jerky or exaggerated. Now the brain and the brainstem and the spinal cord are very valuable pieces of equipment in the nervous system. If they're damaged, they can't be replaced, and so nature has seen fit to protect them with a variety of layers and protective cushions. So this is a cross-section through the cranial vault, and what you can see is that the skin and the scalp form the outer surface, the most superficial layer of the cranium. Beneath that is the periosteum, which is the membrane that surrounds and nourishes the flat bones of the skull, as well as the rest of the bones of the body. Beneath that is a layer of compact, then spongy, then compact boned again. And we talked about the fact that what the spongy bone between the two compact layers does is it distributes any force that's applied to the bone over a greater surface area so that the bone is less likely to fracture. Beneath the last layer of compact bone, you've got a layer of dura mater, which is the outermost protective membrane surrounding the central nervous system. Underneath that is the arachnoid mater. Okay, so you can see the arachnoid mater here. It looks like a little purple sheet in this diagram. These are arachnoid granulations. This is where the cerebrospinal fluid rejoins the general circulation and is returned ultimately to the inferior vena cava, which feeds into the right atrium of the heart. And then beneath the arachnoid mater is the pia mater, represented here by the turquoise colored layer that sits just on top of the gray matter of the brain. And what these three layers do is they cushion and protect the brain, and they also provide a route for blood vessels uh, within and around the nervous system tissue so that we can supply oxygen and nutrients and keep those tissues doing their job. In addition to that, remember that the cerebrospinal fluid covers the outside of the brain as well as filling the inner ventricles, and so the brain actually floats in the cerebrospinal fluid just like a goldfish in a goldfish bowl. And this allows the brain to come into less contact with the bones that overlie it, and this as a result reduces brain damage on a day-to-day basis. If we take a cutaway and look at the other layers, we can see here again the periosteum, and then the meningeal layers made up of the dura, the arachnoid, and the pia mater down here. Again, the arachnoid matter is called such because it has web-like extensions. Arachnoid means of or pertaining to a spider, hence the name of the layer. The pia mater is the internal layer that clings to the surface of the brain and provides a passageway for blood vessels and lymphatics. I will pick up the last component of this lecture in the following block, so stay tuned. Okay, welcome to the last installment of the Brain and Endocrine Lab. This is the block on the endocrine system, so we'll be going through what the endocrine system is, where the relevant organs are located, the hormones that they secrete, and the actions that they trigger. So one of the things that we have to understand about the endocrine system is the fact that the endocrine system is another communication system in the human body, and the reason that we have an endocrine system is because it would be impractical to use the nervous system for every communication function that we need in the body to maintain homeostasis, because it would consume a massive amount of energy, and logistically it wouldn't be practical to wire every cell in the body up to the central nervous system. So the endocrine system is a method of communication between different parts of the body using chemical messengers that are called hormones that are secreted into the bloodstream in response to changes in the body's conditions, and they're carried around the circulatory system by the beating action of the heart, and when they arrive at a target cell, they'll bind to a protein called a receptor, which in the case of water-soluble hormones will be on the cell membrane surface, and in the case of lipid-soluble hormones will actually be inside the cell. And when that happens, it'll produce a change in the target tissue, causing it to perhaps grow, perhaps make a novel gene product, a special protein, or to activate a certain series of enzymes. All of these are possible responses in target tissue. So a cell's function changes in response to a hormone if it has a receptor for that hormone. Just like a key will only fit one lock, a hormone will only bind to a specific receptor. A cell's function is unchanged if the cell lacks the receptor for that hormone, or if the receptor isn't made properly. And so you can see here an example of a couple of different ways that hormones act on their targets. In the case of hormones that are lipid soluble, basically what we would think of as nonpolar or greasy molecules, what they actually do is they slide through the plasma membrane, and their receptors are actually within the cell. The steroid hormone will bind to its receptor, be escorted into the nucleus, and it'll bind to a special part of the DNA called the promoter, which is the gateway to the expression of a particular gene, and that will cause messenger RNA to be produced from that gene, and as a result special proteins will be made by the ribosomes in the cytoplasm, and that will suffice as the response to that particular hormone signal. In the case of water-soluble hormones, The receptor is on the surface of the membrane. When the hormone binds to the receptor, that causes a change in the shape of the receptor, and that triggers a series of chemical reactions inside the cell, which begin with what we call a second messenger. In this case, the second messenger is something called cyclic AMP, which is generated by hydrolyzing ATP and producing this intermediate molecule, which will then go on to activate certain enzymes and cause a response inside the cell that's dependent on what the hormone was designed to do in the first place. And so what happens is that we begin what we call a cascade inside the cell, where the second messenger will activate a downstream protein. That downstream protein will activate another downstream protein, and eventually what will happen is it will either send an activated protein at the end of the cascade into the nucleus, where we'll express novel gene products, or these activated enzymes will produce a product that the cell needs in order to respond properly to the hormone signal. And the reason for this cascade arrangement is so that you can take a very small initial signal and amplify it at each step in the pathway, and the result is that you take a tiny signal and amplify it really quickly, and once you generate now the end product, what happens is that to turn off the cascade, all you have to do is stop the first step in the cascade where the second messenger is made, and then the whole thing shuts off. So you take a tiny signal initially, the hormone binding to the receptor, you amplify it at each step in the pathway, and then once you produce the product that the cell needs to make in response to the hormone, that will turn off the first step in the cascade inside the cell, and the whole pathway will shut down. And so you are allowed a great deal of control, as well as the ability to amplify the signal very quickly. Okay, this is just an outline of the endocrine system, major glands, and what we can do is take a brief tour of the endocrine system by walking through each of the different organs. We can start on the underside of the brain in the diencephalon. Remember that the diencephalon is made up of the epithalamus, the thalamus, and the hypothalamus. The hypothalamus governs an organ called the anterior pituitary, and it does that by producing releasing hormones that make the anterior pituitary produce hormones that talk to the other endocrine glands and cause them to carry out specific functions based on these commands. The pituitary gland, which lies directly underneath the hypothalamus, is actually split into two lobes, an anterior and a posterior lobe. The posterior lobe of the pituitary gland is actually an outgrowth of the brain, and it only makes two hormones, oxytocin and antidiuretic hormone, which are actually synthesized in the hypothalamus and stored in the posterior pituitary. And what these two hormones do, oxytocin is involved in the ejection of milk from breast tissue and uterine contraction, and antidiuretic hormone is your water retaining hormone, so it's important in maintaining blood pressure, blood volume, and fluid balance. The anterior pituitary is controlled by secretions from the hypothalamus called releasing factors that produce trophic hormones that target other endocrine organs. So you could think of the hypothalamus as the commander-in-chief of the rest of the endocrine system, and the pituitary gland as the five-star general. So what happens when the anterior pituitary is stimulated is that there's a series of hormones that are produced that talk to different endocrine organs. So what we can do here is draw some relationships. One of the hormones that are produced by the anterior pituitary is a hormone called thyroid stimulating hormone, or TSH. And what that does is that tells the thyroid gland to produce its hormone. Its hormones are thyroxine and another hormone called T3. Another set of hormones that are produced by the anterior pituitary is called ACTH. Okay, and ACTH targets the adrenal glands, specifically the adrenal cortex down here. So A, C, T, H. And what this hormone does is it stimulates the adrenal cortex to secrete its hormones, which include aldosterone, which is your salt retaining hormone, so it controls electrolyte and fluid balance, and also cortisone and cortisol, which control your blood sugar. Another tropic hormone that is secreted by the anterior pituitary are a set of hormones called gonadotrophins, and they target the gonads. In the female, the gonads are the ovaries, and in the male, the gonads are the testes. And the two gonadotropins that are produced by the anterior pituitary are called LH and FSH, and that those two names stand for luteinizing hormone and follicle stimulating hormone. And what they do is they stimulate the gonads to produce their hormones. In the case of the female, the ovaries produce estrogen and progesterone, and in the case of the male, the testes produce testosterone. So you can see here that these hormones are very important. Other hormones that are produced by the anterior pituitary include HGH, that stands for human growth hormone, and what that causes you to do is to grow, to increase in size, HGH. And then there's another hormone called prolactin, which targets the breast tissue and stimulates milk production. Okay, and then PRL, prolactin. And so you can see that the anterior pituitary is pretty important. Controlled by the hypothalamus, since the hypothalamus is part of the brain, you can see that if we have a problem with the functioning of the brain, we can have a problem with the functioning of the hypothalamus. That can affect the activity of the pituitary, and that can have wide-ranging effects on the functioning of the endocrine system. So this is one of the reasons why in holistic medicine, where we treat not only the physical symptoms of a disease but also the patient's state of mind, often they show a higher rate of success with dealing with the treatment of the disease than simple Western medicine, where we simply give you a drug to alleviate the symptoms. Now, on the backside of the brainstem, opposite the pituitary gland, is a gland called the pineal gland. The pineal gland produces a hormone called melatonin, which controls when we're asleep and when we're awake. Melatonin tends to peak during periods when it's dark out, and it tends to decline when it's light out. So there's some evidence to suggest that this, along with the reticular activating system, is part of what governs our activity levels, so that we're active in the daytime when we can see and do things and enhance our survival by finding shelter and gathering food and so on. And we're inactive when it's dark out because it's difficult to do any of those activities. And if you were active when it was dark out, it's very likely that you could suffer an accidental death, either as a result of, say, walking off a cliff or being consumed by a predator or what have you. You have to think in terms of, a long time ago, before we had modern conveniences like electric light and so on. This is back when we were relying on the sun to provide us our day and night cycles. We didn't have any other source of light. The hypothalamus we've discussed, the pituitary gland we've discussed, the thyroid gland down here is a butterfly-shaped gland that sits right in front of the trachea, and it produces two hormones, T3, which goes by the name of triiodothyronine, and thyroxine, which is abbreviated T4. The 3 and the 4 indicate the number of iodine atoms in each of these hormones. And what these hormones do is they accelerate your metabolic rate, which is basically the speed with which you burn your stored fat. So if you have a lot of thyroxine and T3 being secreted, your body fat percentage is going to go down, and you're going to generate lots and lots of energy. So you'll tend to be lean and mean and have a rather high body temperature and low body weight. Another hormone that's produced by the thyroid is called calcitonin. And what calcitonin does is it lowers blood calcium levels. So calcitonin, T-O-N-I-N. And it does this by stimulating the osteoblasts, which are the bone builders, and this basically takes calcium out of the blood and puts it back into compact and spongy bone, and it increases bone density. The parathyroid gland is a set of glands on the back of the thyroid, and they produce a hormone called parathyroid hormone, or PTH. And what PTH does is it increases blood calcium levels by stealing calcium from the blood, increasing calcium retention by the kidneys, and increasing calcium absorption from the digestive tract. And so calcitonin and parathyroid hormone work together to regulate blood calcium. You have to remember that blood calcium is important not just in maintaining strong bones, but it's also vital in nerve transmission and muscle contraction. So if we don't tightly regulate our blood calcium levels, the nervous system and the muscular system will shut down, and you'll die in very short order. The thymus gland, shown right here, is the site of T cell maturation. Your T cells are part of your body's defense against disease, and what happens early on when you're still a newborn is that immature T cells they migrate to the thymus, and there they become chemically educated so that they can be immunocompetent, basically capable of fighting disease. Once that happens, the thymus, the T cells in the thymus will then move out to the other lymphatic organs, which include things like the spleen, the liver, the lymph nodes, the tonsils, and so on. And then the thymus will slowly begin to involute and eventually become non-functional. The thymus is located behind the sternum and in front of your heart. In the young children, it's quite large. In adults, it's rather tiny because now it's already served its purpose, and we no longer need the T cells to actually occupy this space. The hormones that the thymus gland produces are thymosins and thymopoietins, and they're designed to stimulate the maturation of the T cells, which are, again, a very important part of our adaptive immunity. Now, the adrenal glands are actually two glands in one. The outer layer is called the cortex, and the inner core is called the medulla. Now the medulla is fairly straightforward. The medulla secretes a hormone called adrenaline. A D R E, adrenaline. And what adrenaline does is it coordinates our fight or flight response. Essentially, its actions include the acceleration of the respiratory and the heart rate, an increase in blood glucose levels and blood pressure, a decrease in urine output, dilation of the pupils, and as a result, what happens is that we create conditions that allow you to very quickly deal with a situation in which you're in imminent danger. Okay, that's why we nickname the sympathetic division of the autonomic nervous system the fight or flight division, and its effects are enhanced by the secretions of the adrenal medulla. Now, the outer layer of the adrenal gland, the cortex, secretes a series of hormones that are called corticosteroids, and the outer layer of the adrenal cortex produces our salt retaining hormone called aldosterone. I'll just abbreviate this ALD for short. And what aldosterone does is it causes the body to retain sodium and as a result also retain water. The middle layer of the adrenal cortex produces cortisone and cortisol, which are what we call glucocorticoids. And what they do is they suppress inflammation and they also increase blood sugar levels. So they're what we call glucocorticoids, C-O-R-T there. And then the inner layer of the adrenal cortex produces androgens, okay? And what these are hormones that are very much like testosterone, and so their effects are to increase muscle mass, increase bone density, increase hematocrit, and increase essentially what we would call and reduce your fat content. Okay, it also promotes the production of male secondary sex characteristics such as axial and body hair, deepening of voice, and so on. So, in every human, you've got two sources of sex steroids. Those include the adrenal cortex and the gonads. Okay, it's getting a little crowded here, so let's erase some of this stuff and then go back. Now, the pancreas, right here, okay, the pancreas, is actually a gland that does two jobs in one. It's both an exocrine gland and an endocrine gland. Its exocrine function, and by exocrine gland we mean that the product of a particular gland is secreted through a hose that we call a duct that either squirts its product into another organ or onto the surface of a tissue. So its exocrine function is to produce digestive enzymes. Okay, so digestive enzymes. Which help chemically break down the food that we eat so that we can absorb it across the wall of the small intestine and into the bloodstream where we can use it for fuel. Its endocrine function is to produce two hormones that control our blood sugar levels, and those include insulin and glucagon. And what these two hormones do is they keep your blood sugar levels within very tight limits. Insulin is secreted when your blood sugar levels go up, and its effect is to lower blood sugar, while glucagon is produced between meals when blood sugar levels are low in order to increase blood sugar. So these two hormones seesaw back and forth to keep your blood glucose levels within very tight limits. Now, the last set of endocrine organs that we want to talk about are the gonads. And in females, the gonads are the ovaries, and in males, the gonads are the testes. And the hormones that are secreted by the testes, shown down here, okay, are testosterone. Okay, testosterone, T-E-S-T-E-R. O-N-E. And what does testosterone do? Well, it accelerates sperm production in the male. And we should point out that the gonads are another one of those organs that's both an exocrine and an endocrine organ. The secretory product of the testes is sperm, and the endocrine product of the testes is testosterone. Now, aside from accelerating sperm production, testosterone also increases hematocrit, increases bone density, increases lean muscle mass, uh, and it also reduces overall body fat, in addition to promoting male secondary sex characteristics such as body hair, deepening of the voice, and so on. In the female, the gonad is known as the ovary. There's an ovary right there, right next to a fallopian tube. And the ovaries produce estrogen and progesterone. And what estrogen and progesterone do is they coordinate the ovarian and the uterine cycles in the female. And what we're going to do here now is we're going to sort of tie in our discussion of the pituitary gland that we had a little while ago with the impact on the gonads. Now, in the female, luteinizing hormone and follicle stimulating hormone, which come from the anterior pituitary, both target the ovary. Okay, so let's put LH and FSH up here. Now, what happens is that the secretion of these two hormones by the anterior pituitary is stimulated by the hypothalamus's release of something called gonadotropin releasing hormone, GnRH. The first hormone to be secreted in significant amounts beginning every month once puberty sets in the female is follicle stimulating hormone. And what that will do is that will promote the maturation of about 24 follicles inside the ovary, hence the name follicle stimulating hormone. We're talking about the follicles. Now you might ask, well, what is a follicle? A follicle is basically a house which holds a single egg, and there's thousands of them inside of each ovary of the female. As follicle stimulating hormone acts on the ovary, what happens is it begins to fill with fluid and enlarge, and eventually of those 24 follicles, one will be chosen to essentially move out of meiosis 1 into meiosis 2, the egg that is, and that particular follicle that contains that egg will become so brittle that the stroking action of this sort of frilled edge of the fallopian tube as it strokes the ovary will cause it to burst, will pop, just like an overfilled water balloon. And the egg will come out and be sucked into the fallopian tube. And if there are sperm around, it'll be fertilized and form what we know as a zygote. And at that point, what we've done is we've begun the formation of a new human being. Now, as the zygote progresses in development, what will happen is that one cell will become multiple cells. So let's draw a single cell here. Okay, that's the zygote. And then it'll become multiple cells as it divides very rapidly. And this multiple cell mass is what we call the morula. Okay, so there is the morula. And then as it continues down the fallopian tube, eventually what will happen is it'll transform into a hollow ball of cells called the blastula. So there's our hollow ball of cells. Okay, there's the blastula, the inside of the blastula filled with fluid, and the blastula will float there for a few days, feeding off secretions that come from the uterus before it decides to implant. Now, while all this has been going on, the follicle that was left behind in the ovary from which the egg erupted has begun to transform into a yellow mass called the corpus luteum, which is Latin for yellow body. And this happens under the influence of this second gonadotropin called luteinizing hormone. The luteinizing hormone, as it transforms the follicle into the corpus luteum, will cause it to secrete large amounts of progesterone, which will serve to cause the endometrium of the uterus here, okay, to begin to engorge with blood and nutrients for the blastocyst, so that when it implants, it'll have an opportunity to get nutrients from the mother and continue to develop. Prior to the egg's arrival, the follicle has been secreting smaller amounts of progesterone and also estrogen, which has also thickened the endometrial lining in preparation for the arrival of the blastocyst. Once the blastocyst implants, this will implant into the endometrium. Okay, so endometrium. It'll grow a stalk that'll connect it to the lining of the uterus, and that stalk is called the chorion. The chorion will secrete a hormone called human chorionic gonadotropin, or HCG. And what HCG will do for the first 1.5 trimesters of the pregnancy is allow that corpus luteum to remain intact inside the ovary and continue to secrete progesterone, which will maintain the endometrium long enough for eventually the chorion to transform into the placenta and the umbilical cord. And at that point, the placenta and the umbilical cord will actually take over the job of secreting progesterone to maintain the endometrium while the last stage of development completes, the last 1.5 trimesters. So every pregnant woman has 2 extra endocrine organs. They have a corpus luteum in the ovary, and they have a chorion in the uterus, and they talk to each other through this hormonal axis carried again in the blood. Once the baby's born, what happens is that the amount of human chorionic gonadotropin begins to drop through the remaining 1.5 trimesters. And as the human chorionic gonadotropin disappears, what will happen is that that yellow body in the ovary, the corpus luteum, will eventually no longer be needed, and it will transform into a white mass called the corpus albicans, and will no longer be a hormone-secreting structure. This happens every month in the woman from puberty until an event called menopause, which represents the last egg being ovulated from the ovary, and with that, the last corpus luteum and corpus albicans being created. And at that point, what happens is that the amount of estrogen progesterone produced by the female gonads drops precipitously, and often what this leads to is osteoporosis, because one of the other things that estrogen and progesterone do besides regulating the reproductive cycle is they also help maintain bone mass. So often what we'll do is we'll try and replace these decreased amounts of estrogen and progesterone with hormone that comes either as an oral or as an injectable, and that's what we call hormone replacement therapy, or HRT. So that's pretty much the tour of the endocrine system, and what follows in the next several slides is a list of the different endocrine organs, the hormones that they produce, their mode of action, and their target tissue. So as you look at the handout, which is up on Angel, make note of this list of different endocrine organs, okay, and be able to categorize the major hormones that each one of them produce. So the numbers indicate the number of major hormones secreted by each endocrine organ. Okay, so starting up here, there's the pineal gland, which secretes melatonin. Over here, the anterior pituitary, which secretes human growth hormone, prolactin, adrenocorticotropic hormone, luteinizing hormone, follicle stimulating hormone, thyroid stimulating hormone, and there's one other, 6, 7, 8. Give me a second here and it'll come to me. I think that's it. Down here, the thyroid gland secretes thyroxine, T3, and calcitonin. The parathyroid gland secretes parathyroid hormone. The thymus secretes thymosin and thymopoietin. The adrenal cortex and adrenal medulla secrete a host of hormones, the mineralocorticoids, the glucocorticoids, and the androgens, as well as epinephrine and norepinephrine by the medulla. Okay, the main mineralocorticoid being aldosterone, the main glucocorticoids being cortisone and cortisol, and the androgens, of course, a host of sex steroids that are an awful lot like testosterone in their target and in their structure. And then finally the medulla, which secretes epinephrine and norepinephrine. The islet cells of the pancreas secrete insulin and glucagon. The ovaries secrete estrogen and progesterone. Of course, the testes secrete testosterone. So as you look at each of these hormones, that was my problem with the pituitary. I forgot that the posterior pituitary also secretes oxytocin and vasopressin. So you can see here on the list the gland, the hormone, the chemical structure, okay, the major actions, and what regulates it. And so in case you're wondering, yes, you need to know this information. And this is simply a diagram showing you what is targeted by each of the anterior pituitary hormones, okay, the target of choice, and the posterior pituitary as well, secreting oxytocin and antidiuretic hormone, which remember, oxytocin stimulates milk ejection from the breast and uterine contractions, while antidiuretic hormone is our water retaining hormone, so it reduces urine output and increases blood volume and blood pressure. And then all these other hormones, okay, the trophic hormones, ACTH, TSH, thyroid stimulating hormone, FSH and LH, and then prolactin, which stimulates milk production in the breast, and human growth hormone, which stimulates the growth of both the skeletal system and the soft tissue connected with it. And what you're looking at here is just the underside of the brain model, and this is where the pituitary gland would be if it were still present. Very often when you remove a brain from the skull, as in our sheep brain, whose dissection we'll see at the end of this presentation, what will happen is that gland will actually pop off, and the only thing that you'll see connected to the underside of the brain is the stalk that attaches it to the hypothalamus, called the infundibulum. You can see here the olfactory bulbs. There's the optic chiasm and optic nerve, and of course here is the medulla oblongata and the pons, and you can see the various cranial nerves radiating out from the brainstem as well. There, of course, is the cerebellum, the occipital lobe, the temporal lobe here, and of course the frontal lobe. Looking here, okay, talks about the pineal gland and the hypothalamus, the hormones that they produce, the chemical class that they belong to, and their major actions and how they're regulated. And here you can see again just a further discussion of how the command structure works in the endocrine system, where the hypothalamus monitors blood supply and body conditions and releases hormones called releasing factors, which in turn stimulate the production of trophic hormones, thyroid stimulating hormone that targets the thyroid gland, adrenocorticotropic hormone that targets the adrenal cortex, and follicle-stimulating and luteinizing hormone, which target the gonads. And in the male, those are the testes, and in the female, that's the ovary. Okay, so this is how the command structure works. And as you may well have surmised, an error in the hypothalamus or in the anterior pituitary is going to cause malfunction of the organs that are downstream of it, and the result is going to be endocrine disorder. So this is one of the sources of endocrine diseases, is a malfunction in the hypothalamus or the anterior pituitary, where it's either underactive or overactive, and is not listening really to the body's changing conditions, and causing now everything downstream of it that it controls to produce inappropriate amounts of hormone, and as a result have wide-reaching systemic effects on the body. Another way that the endocrine system can malfunction is that you can have an error in these targeted glands themselves, where they can either over or under secrete their designated hormone, and that will lead also to problems in endocrine function. Here you can see on the brain model the location of the pineal gland. This is the choroid plexus. This is the corpus callosum. This is the hypothalamus. This would be the infundibulum, and the pituitary gland would be right here. It's not shown actually in this brain model. And remember that the pituitary has two lobes, the anterior pituitary in the front, which is glandular tissue that actually comes from a structure early in embryonic development called the pharynx, and the posterior pituitary, which is actually an outgrowth of the hypothalamus, which stores oxytocin and vasopressin, which are manufactured in the hypothalamus and released when they're needed. You can also see here the cerebellum, this white tree-like structure, the arbor vitae. You can see the medulla oblongata, the pons, the midbrain as well. There is the thalamus, okay? And then this structure here is called the cingulate gyrus. It's part of the limbic system, our emotional brain. And you can also see again the occipital, the parietal, and the frontal lobes in this diagram. This is just a list of the hormones made by the thyroid, the parathyroid, and the thymus. Again, they give you the chemical class, they show you the major actions, and they show you the hormones that regulate them. And here you can see the position of the thyroid gland right below the larynx. It has two lobes connected by an isthmus, okay? And again, its major hormones are thyroxine, T4, triiodothyronine, T3, and calcitonin. And then on the back of the thyroid are the four parathyroid glands, which secrete parathyroid hormone. Okay, so you can see again the location. We're looking at the front of the larynx and the trachea here, and there's the thyroid gland. Occasionally, when the thyroid gland over or under secretes hormones, it will enlarge, and this forms a mass under the chin called a goiter. You may have seen that in the slides in the PowerPoint from the lecture component on the endocrine system. Up here, this is the hyoid bone. This is the thyroid cartilage. Down here is the cricoid cartilage, and these are the tracheal cartilages that hold the airway open as you breathe, just as a way of showing you what you're looking at here. And then you can see here the carotid arteries that bring blood up to the brain, and then drain into the brachiocephalix, into the aorta, on the way back, completing the circulatory circuit. So if we wanted to trace blood going to the brain, it would go like so. So let's imagine here the blood gets pumped through the aorta, it comes up through the brachiocephalic artery, it heads through the common carotid, and from here we head up to the brain. Okay, and same on the other side. Okay, like so, up to the brain. And then just by way of being informative, the trip back is assisted by the internal and external jugular veins, which drain into the superior vena cava, which dumps into the right atrium of the heart. So that completes the circuit of blood to and from the brain. We'll touch on that when we get to the chapter on the circulatory system, but I just wanted you to know exactly what you're looking at in this diagram. And here you can see our model of the larynx, okay, the voice box. It's called the voice box because if we look down into the larynx from the top, we would see vocal cords, and that's how we talk. As air moves past our vocal cords, they vibrate and they make sound. This is the hyoid bone. This is the thyrohyoid ligament. There's the chrysoid cartilage, the thyroid cartilage, and there, of course, is the thyroid gland. Okay, and then if we look at the back of the thyroid gland, those are the parathyroids. Okay, you can see them embedded into the thyroid gland. Remember that the parathyroid glands secrete parathyroid hormone, which has the desired action of elevating blood calcium levels. And you can see here how it is that this is triggered. As blood calcium levels drop, the parathyroid glands are stimulated. They will generate parathyroid hormone secreted into the bloodstream, and this will cause an increase in osteoclast activity. Remember that the osteoclasts are the bone dissolving cells, and so what they do is they tear apart compact bone, releasing calcium into the bloodstream and elevating blood calcium. On the flip side, calcitonin is responsible for causing blood calcium levels to drop. That's secreted by the thyroid gland. So what happens here when we have changes in blood calcium level is that calcitonin and parathyroid hormone will either be secreted in increased or decreased amounts depending on the body's needs. So let's say, for example, that we have a rising blood calcium level. This will stimulate the thyroid gland to secrete calcitonin. Calcitonin will stimulate osteoblast activity in the bone. Calcium salts will be deposited. Bone density will go up. Blood calcium level will go down, and we'll head back to normal calcium concentrations in the blood. If, on the other hand, we have a drop in blood calcium levels, we will stimulate the parathyroid glands to secrete parathyroid hormone. This will increase osteoclast activity, which will result in more calcium being released into the bloodstream, and now calcium levels will go back to where they were before we originally saw them drop. So these two hormones work together. And again, this is just a list of the hormones of the adrenal cortex and medulla, the gland that secretes them, the hormone that they make, the chemical class that the hormone belongs to, their actions, and how they're regulated. And so here you can see when we're talking about the adrenal medulla and the adrenal cortex, we're actually talking about two glands in one. When the sympathetic division of the autonomic nervous system is active, when you sense a dangerous situation, what will happen is that there's a two-neuron chain that heads out from the gray matter of the spinal cord through the ventral root, and these motor efferents, okay, these efferent neurons will send an electrical signal to the core of the adrenal gland, and this will cause it to release massive amounts of epinephrine and norepinephrine, also sometimes called adrenaline and noradrenaline, into the bloodstream. And this will cause an increase in heart rate, an increase in respiratory rate, the dilation of the pupils, an increase in blood glucose concentration, an increase in blood oxygen concentration, and also reduce the urine output, thereby increasing the blood volume and the blood pressure. And this is all to deliver more oxygen and nutrients to the skeletal muscles so that we can deal with the situation at hand and get ourselves out of danger. If we look at the adrenal gland as two glands in one, we can also see that when we're under the influence of the fight or flight division of the autonomic nervous system, the sympathetic division, we also stimulate the adrenal cortex via the release of adrenocorticotrophin releasing hormone from the hypothalamus, which stimulates adrenocorticotrophic hormone release from the anterior pituitary. That's carried in the blood to the adrenal cortex, and that results in the secretion of cortisone and cortisol as well as aldosterone. And if we look at what the cortisone and cortisol do, what they do is they mobilize sugar so that we can have a higher glucose content in the blood. They also suppress the response of the immune system, so inflammation is suppressed. The aldosterone causes retention of water and sodium by the kidneys, which increases blood volume and blood pressure. And the net result is that we have a whole lot of blood with a whole lot of oxygen and a whole lot of fuel in it getting to the skeletal muscle and helping us get out of danger. So the two uh, broad segments of the adrenal gland are stimulated at the same time when the sympathetic division of the autonomic nervous system has really kicked into gear. You can see some of the other effects of epinephrine listed here, okay, among them bronchodilation, okay, so that we can increase the amount of oxygen getting into the lungs, and we have changes in the pattern of blood flow. So we basically decrease blood flow to the kidneys and increase blood flow to the muscles, and of course we have an increased basal metabolic rate, so that we burn the fuel in the blood more quickly, turn it into ATP, and use that ATP immediately to do something, okay, to perform some kind of an action. Here you can see the location of the pancreas. It's nestled between the first segment of the small intestine called the duodenum and this gland up here called the spleen, which recycles red blood cells but also has a role as a lymphatic organ in that it screens the body's blood plasma and tissue fluid for pathogens and alerts the immune system to their presence. And remember that the pancreas is a switch hitter. It both has an exocrine function, the production of digestive enzymes which are secreted into the pancreas to break down the food that we eat so that we can get it into the bloodstream across the lining of the small intestine and use it for energy. And also its endocrine function, which is the secretion of two hormones that act to regulate blood sugar levels. And the way they do that is that when blood sugar levels begin to rise, the pancreas is stimulated to release insulin, which causes cells to take the insulin up and use it for energy, and also causes the liver to store the sugar in the blood as glycogen or to convert it into fat. And so what happens under the influence of insulin is that the blood glucose levels begin to drop and we head back down to our normal glucose balance. In between meals, when blood glucose levels begin to drop below what we would consider an optimal level, the stimulation of glucagon um, will cause the breakdown of glycogen in the liver to produce simple sugars, glucose in the blood, and will also stimulate the liver to engage in gluconeogenesis. That means literally the formation of new sugar from things like amino acids. And as a result, the blood glucose level will rise as these compounds begin to enter the bloodstream, and blood glucose will then head back to its normal range. So these two hormones act together to keep our blood glucose levels fairly tightly controlled. And then finally, the gonads, again listed here, the hormones that they produce listed, their chemical class. They're all lipid soluble hormones, meaning they're nonpolar, which means their receptors are inside the cell instead of at the cell membrane surface. Their major actions, shown here, target the reproductive system and also secondary sex characteristics, and they're all regulated by follicle stimulating and luteinizing hormone made by the anterior pituitary. Those, remember, are the gonadotropins. And so here you can see the ovaries and the testes, okay, their location by way of orientation. What else are you looking at here? That's an ovary. That's the end of the fallopian tube, which leads into this organ here, the uterus, okay? The inner lining of the uterus is the endometrium. The middle lining is the myometrium, and the outer lining is the perimetrium. It's the endometrium that nourishes the developing embryo and fetus by engorging with blood and nutrients during the 3 trimesters of pregnancy. You can see here the cervix. This is the vagina, okay? And this, of course, is the anus and the rectum. This is the urethra. This is the bladder. Okay, so you can see that the uterus sits right on top of the bladder. And this is the pubic symphysis, which is the joint, the amphiarthrotic joint in the front of the coxal bones where the two pubis come together, and this is the joint that has to be rendered more flexible when the baby is going to head through the birth canal. So what happens is that there's a hormone called relaxin that sort of softens this joint up a bit and allows that large cranium to pass through this very tiny opening so that we can facilitate birth. And so what you should do with the information in the handout, in the lecture, and in these podcasts is you should go through and with each of these hormones list the gland that makes them, list the target tissue, and list the effects. Okay? And if you like, making flashcards out of this is an excellent idea. So do that for the hormones listed here, here, and here, and here finally. And what I will follow with is a couple of things. I will show you a couple of videos on the tail end of this presentation that talk to you about how it is that the hormones actually do their job, how they actually produce responses in their target cells. And then I'll show you a brain dissection so that you can see what a sheep brain looks like as it's taken apart, and you'll know some of the major structures. And hopefully we'll get to do that sometime next week before the practical, so you can get a good look at what an actual preserved brain looks like. And remember that we're going to have the exam, the lecture exam, on Tuesday, and the lab practical on Thursday. Okay, so I want you to be fully prepared for that. Also note that the practice test is already up on Angel for you to use as a study guide. And remember to take notes as you review the lecture and lab podcasts over this material so that you can improve your performance on the exam. Okay, so stay tuned for the dissections and the videos on hormone action. And also, it's probably a good idea for you to go into the video content portion of Angel and watch the videos that are there on the endocrine and the nervous system, as they'll also be very useful in preparing you for the upcoming exam. Okay, so I'll see everybody in class.
We have a sheep testicle. These two big bulgy things here in the front are the olfactory bulbs, which are used for sense of smell, to smell stuff. Okay. These two stalks here are the optic nerves, used for vision. And the point where the optic nerves cross is known as the optic chiasm, and they lead to the sides of the brain. This general area here is where the hypothalamus would be. That area, but it's removed.
And then
the stalk where the hypothalamus, where the pituitary gland connects, is known as the infundibulum.
Can see it.
It'll be this little area here. It's a stalk that connects the pituitary gland. And right behind it is the mammillary body. They make up the butt, or the superior caligula from a dorsal view. This bulge here would be the pons. Further down would be the medulla, and this would lead into the spinal cord. Is the brain from the top view.
Voice.
In the front is the frontal lobe. Who would have guessed that? And on the sides, you have the parietal lobe. And at the very back, you have the occipital lobe. And on the sides here, you really can't see it, then you have the temporal lobe.
And that includes that big...
All of that general area there. Okay. So all the folds in the brain are known as gyri or gyruses. These, folds.
Oh, sorry.
And the stuff in between them are known as sulci. That's where capillaries and whatnot run through.
Sulci, right?
Sulci.
Cool.
Now there are a few major ones. This one that separates the brain into two lobes, that's the longitudinal sulci.
Fisher, no?
Fisher, whatever, longitudinal shit.
This would be like, American for
Oh.
kids
So
on the age of
around this general area here
Central?
is the central sulcus.
Which
it divides
divide?
the frontal and parietal lobes.
Okay.
And from the side, we have the lateral sulci, which divides the parietal and temporal lobes. Okay?
Cool.
Okay. And on the human brain, which you can't see here, we have something, a fifth lobe known as the insula.
Which
it's
is?
between...
It's inside.
In there.
Cool.
It'll be like, in there.
We got the point.
In there.
Should I videotape you together? No?
What else do we need to know? Cerebellum, this front part here, cerebrum,
Mm-hmm.
cerebellum, spinal cord.
Mm-hmm.
Now if you pull back the cerebellum,
Oh, okay, here, you need help?
yeah, in here would be the pineal gland.
Which you can't really see in this camera.
It's not really visible, but take my word for it, it's there.
Like in the asshole?
It's in there, the pineal gland, I just poked it. Okay. Then you also have the superior caliculi and the inferior caniculi. They look like a butt, apparently.
Mm-hmm.
Superior on top, inferior at the bottom. And the cerebellum has 3 hemispheres, even though that doesn't really make sense.
Mm-hmm. You can turn it back?
These on the side, and the one in the middle
Vermis?
is known as the vermis.
That's it?
Yep.
What about pineal gland body? That's the piece-shaped thing?
Pineal gland, this is the brain from a medial view, aka cut in half.
So this is the corpus callosum,
The top part?
the top part. The gap right here is the lateral ventricle.
You really gotta poke deep in there to find it.
And the membrane covering it is the septum pellucidum,
Which you don't really see.
which you can't really see right here. If you pull out some of this brown matter right here,
Oh,
you can see it right there.
So blurry.
That is the choroid plexus, some capillaries. The bottom right here is fornix. Can't see this
that
area
circle
right
thingy.
here. The circle is the thalamus. The cingulate gyrus is this little division kind of touching the corpus callosum, so just right on top of it right here. The cerebellum, obviously, and you have all the white matter is the arbor vitae, and all the brown matter is the folia. This gland right here, this little tiny one, is the pineal gland.
Looks like a pea.
Yeah. This gap right here is called the cerebral aqueduct. Then you have, going from here down here, this is the fourth ventricle.
Not quite.
No, it's
The fourth
Well,
ventricle is
it
the
kind of
gap
comes
inside
around
of
here, and then it comes all the way down here through the central canal, and then out by the spinal cord. That's
The endocrine system is composed of ductless glands that secrete hormones, chemical messengers that control homeostasis. The hypothalamus controls the secretions of the pituitary. Nerve cells in the hypothalamus send releasing hormones to anterior pituitary to release hormones. Hormones are growth hormone, TSH, ACTH, MSH, FSH, LH, and prolactin. Hormones produced by the hypothalamus are stored in the posterior lobe of the pituitary. Hormones are ADH, increases the reabsorption of water, and oxytocin contracts the pregnant uterus. Pituitary gland is called the master gland because it controls the function of other endocrine glands. T3 and T4 regulates metabolism of nutrients for normal growth and matures the nervous system. Calcitonin lowers blood calcium and phosphates. Parathormone raises the blood calcium to normal levels. Medulla secretes adrenaline and norepinephrine that prepare the body for stressful situations. Cortex secretes aldosterone, regulates sodium reabsorption and potassium excretion in the kidney. Cortisol reduces inflammation. Androgens stimulate male sex characteristics. Testes produce testosterone. Ovaries produce estrogen and progesterone, hormones necessary for development of reproductive systems and secondary sex characteristics. Thymosin is necessary for the development of T lymphocytes. Melatonin regulates the body wake and sleep cycles and inhibits the function of the reproductive system. Serotonin acts as a neurotransmitter and vasoconstrictor. Islets of Langerhans produce insulin and glucagon that regulate the blood glucose level essential for energy.
Hormonal communication generally begins with a part of the neuroendocrine system receiving sensory information and reacting by issuing a command to the body in the form of a hormone. In this example, dehydration is detected by osmoreceptors in the hypothalamus, which then directs or stimulates the posterior pituitary gland to release antidiuretic hormone, ADH. The next step is to transport the hormone molecules to target cells. This is typically done via the bloodstream. Here, ADH is transported through the bloodstream to the kidneys and blood vessels. When hormone molecules reach target cells, they will bind to matching receptors on those cells, and the hormone receptor complexes will trigger changes in the target cells. This causes the kidneys to reduce the urine volume output, thereby increasing water retention and countering dehydration. In blood vessels, vasoconstriction is increased, leading to higher blood pressure and thus countering the blood pressure drop caused by dehydration.
Peptide hormones influence their target cells without actually entering them. They bind to receptors on the target cell's plasma membrane. This will trigger one of a variety of responses. For example, epinephrine from the adrenal medulla binds to a receptor on the membrane of a liver cell. Epinephrine's effect on this cell will be the breakdown of glycogen to glucose. The binding of epinephrine to the receptor activates the enzyme adenylate cyclase, which converts ATP to cyclic AMP. Cyclic AMP acts as a second messenger and sets off a cascade of reactions that activate a set of enzymes. In this cell, the enzymes catalyze the breakdown of glycogen to glucose, and glucose can be released from the cell.
Steroid hormones are not water-soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins. Thyroxine is a water-insoluble hormone that is brought to the target cell via a protein carrier. Because it is lipophilic, thyroxine can easily pass through the cell membrane. Thyroxine contains 4 iodines and is often abbreviated T4, tetraiodothyronine. The thyroid gland also secretes smaller amounts of a similar molecule that has only 3 iodines, called triiodothyronine, T3. Both hormones enter target cells, but all the T4 that enters is converted into T3. Thus, only the T3 form of the hormone enters the nucleus and binds to nuclear receptor proteins. The hormone receptor protein complex, in turn, binds to the appropriate hormone response elements on DNA. The binding of the hormone receptor complex has a direct effect on the level of transcription at the site where it binds. The messenger RNA, mRNA, produced then codes for specific proteins.